and thank you for joining Cellular News with our special broadcast of the presidential campaign of the cell. Today, we will follow the lipid processing party from directly where the organelles and their functions are working. The lipid processing party has been in full run and function since the beginning of the cell, and they aren't stopping anytime soon. Members of the lipid processing party, which include the smooth ER, Golgi apparatus, and vesicles, have been out and about, making the production of macromolecules, their modification, and transportation functioning as efficiently as possible.
The smooth ER, the Golgi apparatus, and the vesicles all play an essential role in the processing, modifying, and transportation of lipids, which is an important macromolecule needed to sustain cellular life. However, lipids are just one of the many macromolecules these organelles transport. Some examples are proteins and carbohydrates.
And now to focus in on specifically, we turn to Hannah with the smooth ER.
Thank you, Eva. My name is Hannah Redmond, and I'm a reporter from the Cellular News following the smooth ER from the lipid processing party. The smooth ER is a long network of folded tube-like structures, or pipelines. The interior is known as the lumen. The smooth ER has no ribosomes, which distinguishes it from its neighbor, the rough ER. The smooth ER regulates and releases calcium ions, and as the factory of the cell, it synthesizes steroids, lipids, hormones, and proteins. It produces and manufactures what the cell needs when it needs it.
Cellular membrane! Vote cellular membrane! We allow the good stuff to come in!
Yeah, but you also let the toxins come in, which is why we have to do a process called detoxification to process those toxins. The smooth ER is very beneficial to the function of the cell because as the other organelles do produce the necessary products, without the lipid processing, they would not have the means of transporting them throughout the cell. It puts the products into cisterns and transports them. And on to Natalie with Golgi apparatus.
Hi, my name is Natalie Hoffman, and I'm a reporter for Cellular News. Today we will be covering the Golgi apparatus. The main function of the Golgi apparatus is to be responsible for handling the macromolecules that are required for the proper cellular function. It processes and packages these macromolecules for use within the cell or for secretion. Primarily, the Golgi apparatus modifies proteins that it receives from the rough endoplasmic reticulum. However, it also transports lipids to vital parts of the cell and creates lysosomes. The Golgi apparatus has a function that is made of cisternae, which are flattened stacks of membrane. These cisternae help proteins travel from different points of the cell using enzymes. In order to modify a macromolecule, cisternae enzymes need the addition of carbohydrate and phosphates to properly label each protein for its ultimate destination. These carbohydrates and phosphates are received by the Golgi apparatus through nucleotide sugars delivered to the organelle from the cytosol. The Golgi complex is made of several flattened membrane sacs, but can be ultimately divided into two sections, the cis-Golgi and the trans-Golgi network. The cis-Golgi functions as the receiving end for newly synthesized proteins from the lumen of the endoplasmic reticulum. Vesicles containing proteins from the ER merge with the cis-Golgi, allowing the proteins to enter the Golgi complex. As the cis-Golgi receives proteins from the ER, the protein then begins their modification, moving along membrane to membrane towards the TGN, or the trans-Golgi network. At the other end of the Golgi complex, the newly modified protein arrives at the TGN, where it is then sent off to different parts of the cell via transport vesicles. And finally, the Golgi apparatus is essential and beneficial to cellular function because it acts as a warehouse for receiving, sorting, modifying, and shipping macromolecules that are essential for cellular function. And now to Angeli with vesicles.
Thanks, Natalie. Now onto the segment about vesicles. In cell biology, a vesicle is a small structure within a cell consisting of fluid enclosed by a lipid bilayer. Vesicles form naturally during the process of secretion, or exocytosis, uptake, phagocytosis, and endocytosis, and transport of materials within the cytoplasm. They may be prepared artificially, in which case they are called liposomes. If there is only one phospholipid bilayer, they are called unilamellar liposomes, vesicles. Otherwise, they are called multilamellar. The membrane enclosing the vesicle is also a lamellar phase, similar to that of the plasma membrane, and vesicles can fuse with the plasma membrane to release their contents outside the cell. Vesicles can also fuse with other organelles within the cell. Vesicles perform a variety of functions. Because it is separated from the cytosol, the inside of the vesicle can be made to be different from the cytolytic environment. For this reason, vesicles are a basic tool used by the cell for organizing cellular substances. Vesicles are involved in metabolism, transport, buoyancy control, and enzyme storage. They can also react as a chemical reaction chamber. Vesicle transport. As soon as modified proteins reach the Golgi network, or the TGN, the proteins are separated into several different transport vesicles for different areas of the cell. So there are different types of vesicle types. Exocytosic vesicles are vesicles that contain protein that are to be sent outside of the cell membrane. These vesicles fuse with the plasma membrane, releasing their contents outside of the cell. This process is called constitutive segregation. Secretory vesicles. These vesicles are also to be sent outside of the cell membrane. However, what differs these from exocytosis vesicles is that secretory vesicles need a signal before they can be released. When the signal is given, they will fuse with the plasma membrane to release the contents. This process is called regulated secretion. Lysosome vesicles are vesicles that contain proteins that are sent to the lysosome to be digested. The vesicles benefit the human body by transporting vital necessities throughout the cell.
Vote chloroplast! Vote chloroplast! We're sweet.
Excuse me, without us, you're just a pile of sugar going nowhere. Sorry for that interruption. Back to you, Eva.
Hi. Our slogan is "We transport so you can live." This means that you may produce important macromolecules to sustain cellular function, but without us, you'd go nowhere. So remember, we transport so you can live.
I fly like people get high like planes. If you catch me at the border, I got visas in my...
Skeet, skeet.